Hello, my name is Jakob Hoglund, and I will today talk about conservation genomics of two endangered species in Sweden. These two species are the clouded Apollo that you will see on the left throughout this presentation, and the green toad that you find on the right. Both these species have been part of the ERGA pilot project, and we have assembled draft assemblies for these two species that you see some of the stats on top of these two tables. However, throughout this talk, I will very rarely use these two assemblies because we have been a bit impatient and used shortcuts to get some useful information regarding conservation of these two species. The problem for these two species can be illustrated by the two distribution maps you see below the pictures of the species, and that is that these are rare species found in Sweden, and they are the populations are small and fragmented and far apart from one another. At the bottom of the slide, you also see the sequencing approaches we have been using for these two species. We have been using resequencing approaches for both of them to get population genomic data. In the case of the clouded Apollo, we have mainly relied on mapping to a closely related species called the Apollo butterfly, or Parnassus apollo, which is a closely related species to the clouded Apollo. And in the case of green toad, we have used the DD-RAD protocol and used the de novo assembly using the program STAX. I said that the two species sort of face similar conservation concerns in my country, and you can see these two illustrated in these two sort of maps of Sweden, where the clouded Apollo is found in three different areas, one up here in central Sweden, one area down here, and one in the southern part. There is also population kept at the zoo called Nordens Ark in western Sweden, and these populations have also, individuals from these populations have also been used in this project. We used a very similar protocol when it comes to the green toad, where you see that there are some scattered populations remaining here in the south of Sweden. And we have also included two populations from Poland, two from Denmark, and also a zoo population in this study. So to some results, the first thing we did was to look at population differentiation. And if we start with a butterfly, you see that we have made a PCA here, and that along the first axis, you see that this population here from the north of Sweden is clearly differentiated compared to the ones at the zoo and in southern Sweden, or from mid Sweden. And you also see this in these plots down here, where we look at ancestries using the program admixture. We also see that in the case of the toad, we also find population differentiation. So on the first axis, we find the Polish populations over here, and the Scandinavian, Danish and Swedish populations over here that are spread out along the second axis. What you also see from these two slides here is that the population kept at the zoo is very similar here in this case, that the zoo population and the population in South Sweden are very similar, and that should come as no surprise because the zoo population was founded with individuals from this population. And the same goes for the population for the toad species, where the zoo population was founded from one of the populations in southern Sweden, and these are this blue population here, which is similar to the two samples that we have from the zoo population. So what we do find is that the different populations are clearly differentiated from one another. What you would expect if these populations have been kept or have become fragmented and isolated recently. When we look at diversity, we have done some diversity estimates here. You find these tables that there are different diversity estimates on top. And what we can conclude from this is that we had some difficulties with the butterfly species in mapping to a closely related species because this butterfly genome turned out to be full of repeats and to be unusually large for a butterfly. So we have, in the case of, to be sure that we are really looking at a true polymorphism, we confined our analysis to protein coding genes. But when we did so, we couldn't find any, or we found very similar diversity estimates among the four populations, or the three natural populations and the zoo population. So in this case, this table indicates that we redid this with the mapping to the reference genome from the same species from the clouded Apollo. And here we use all the sites. And what can be noted here is that all the estimates of Tajima's D here, which indicated here that they were negative, they turned out to be positive in the case when we mapped it to the entire genome and the reference genome from. So diversity estimates differed a little bit depending on whether or not we used related species or we used the same species. When it comes to the toad, we can't make any, this is a de novo assembly and we have not had time to map it to the reference genome, but what we see here and what we can conclude is that two continental populations from Poland have higher diversity estimates regardless how we calculate them as compared to the Swedish population. So we can conclude here in the case of the toad that Scandinavian populations seem to harbor less genetic variation than the continental populations. We were also able to look at crude estimates of inbreeding. In the case of the butterfly, we used calculated kinship coefficients. And here you see that somewhat surprisingly, the smallest population in terms of number of butterflies actually had the lowest inbreeding coefficient estimates, whereas the northern population, that is at present the largest, actually had the largest in kinship coefficients, indicating higher degrees of inbreeding at least in the past in the northern population. When it comes to the toads, we see that the estimates vary quite a lot. So some populations are very low. In this case, we use a similar estimate of inbreeding. In this case, we use relatedness. And note that in this case, kinship coefficients can go up to 1, whereas relatedness coefficient can have a maximum at 0.5. But what we can conclude from here is that different populations vary quite a lot in degree of inbreeding. And the least inbred populations are the ones at a few sites in Skonia and the Polish samples, one of the Polish populations, whereas some populations have rather high inbreeding levels. Finally, we were able to look at the mitogenome of these two species. And in this case, we were able to, in the case of the butterfly, we were able to link that to old previously published data. So all the sites or all these populations that you find from Finland down here to Europe is from a study published in 2008, and we have added these three populations. And what this indicates is that since the last glaciation, Sweden has been recolonized from two different areas or from two different directions. One route from the east and establishing this population in the north, whereas these two populations in the south have been probably established via a colonization route from southwest. In the case of the frog, what we see here is that the mitochondria in this species, the green toad here, actually have two different kinds of mitochondria. There are, of course, more diversity within each of these two categories, but there many of the toads in the green toad in Europe has a mitochondria which is similar to this closely related species called Citibundus. And this is probably due to mitochondrial surfing or an integration of mitochondria into this population. And what's interesting is that most of the populations up here in the Baltic and in southern Sweden seem to have this Citibundus mitogenome. This has been a quick survey of the kinds of analysis you can do and which are of interest to conservationists and people working in the actual conservation of these species. And conclusions so far, and I should stress that these conclusions are based on analysis mainly without using the long read reference genomes that we indeed are in possession of today. But the analysis shows strong differentiation of Swedish populations, as expected, the fragmented and isolated populations with limited gene flow. Diversity estimates are possible. Swedish toads less diverse than continental Polish. Inbreeding estimates are blunt, but they show differences among populations. We have mitogenes that indicate differential recolonization routes in the case of the butterflies and mitochondrial progression in the case of the toad. Genomic data can advise conservation actions, for example, which populations to admix, which have the lowest diversity, which are most inbred, and so forth. Finally, I want to sort of just throw in this sort of red herring. We can reach quite good conservation conclusions based on mapping to closely related species or de novo assembly of DD-RAN data. So I'm just throwing this out to the rest of you. Are high quality reference genomes really needed for conservation? Or can we actually go on and do some rough estimates based on less stringent data? I leave that to the rest of you. Thank you very much.